Hello. In this recording, we will look at two groups of organisms that together are informally referred to as protochordates, or invertebrate chordates. They are sea squirts, or tunicates, and lancelets. Neither group is common or well known, but they are important in biology because they help us to understand the early evolution of the phylum to which we belong. These organisms are included in the Invertebrates 3 laboratory exercise. Sea squirts and lancelets are included in the phylum Chordata, the same phylum to which vertebrates are assigned. There are 4 derived characters shared by all chordates at least sometime in their life cycle: a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a muscular postanal tail. Protochordates are placed into 2 subphyla. This is a new classification category for us. Subphylum falls between the phylum and class levels in the taxonomic hierarchy. Lancelets are placed in the phylum Cephalochordata. Tunicates, or sea squirts, are put into the subphylum Urochordata. In the past, all other chordates were included in a subphylum called Vertebrata, but molecular evidence and the desire to make all groups monophyletic have prompted taxonomists to revise this system, as we will discuss in lecture. Let's look at resources for learning about lancelets. Your textbook has useful information about these organisms in the chapter on vertebrates. Here is the model of a lancelet that is available to you in the laboratory. The anterior is on the left. Observe the overall shape. It is this that gave rise to the common name lancelet and to the no longer used genus name Amphioxus, which means pointed at both ends. The anteriormost structure is the rostrum. The tentacle-like structures are buccal cirri, which surround the mouth. The light-colored ridge on top is the dorsal fin, and the fins on the posterior end are called caudal fins. Notice the V-shaped structures on most of the body's surface. These are myomeres, also known as segmental muscles. They are visible through the thin epidermis of the lancelet. The arrangement is similar to that seen in fish and provides evidence that the earliest chordate had a segmented body, as indeed did the ancestors of annelids and arthropods. In addition to the parts that are present, notice the structures that are absent, as compared to fish. There are no eyes, although there are simple eye spots on the nerve cord for light detection only. There are no paired appendages, such as pectoral fins or pelvic fins. Although lancelets can swim weakly, they are filter feeders that spend most of their time burrowed into the sandy bottom of shallow coastal waters. Let's remove the side of the lancelet model and observe the internal organs. The dark pink layer just beneath the dorsal fin is muscle, part of the myomeres. Ventral to the muscle, the light pink structure is the nerve cord. All chordates have the nerve cord in the dorsal side of the body. Notice an eye spot near the front of the nerve cord. As mentioned earlier, this can detect the presence or absence of light, but cannot form images. The green line represents the notochord, the structure that gives this phylum its name. The notochord is a semi-flexible rod that stiffens the body and provides resistance to the effects of muscular contraction, making coordinated movement possible. In vertebrates, this structure is replaced by the vertebral column during early development. The mostly yellow structures constitute the digestive system. Let's identify the main parts. The orange-colored space is the buccal cavity. The narrow region is the velum, We will examine this more closely in a moment. This large region is the pharynx, with its gill bars and gill slits. As we will see shortly, a water chamber known as the atrium surrounds the pharynx. A short, narrow passageway, the esophagus, leads into the stomach. This narrow tube is the intestine, which ends at the anus. Now, let's take a closer look at the anterior portion of the digestive system. Note again the buccal cirri, the buccal cavity, the velum, and the pharynx. The lancelet does not have jaws, nor does it have a muscular pharynx that could expand to suck in food, as we saw in earthworms. Instead, the organism must depend on cilia to draw water and food into the digestive system. At the front of the velum are a number of grooves collectively known as the wheel organ. These structures, as well as the inner surface of the pharynx, are covered in cilia. In addition, a layer of mucus is secreted on these surfaces. Beating cilia set up a current that draws in microscopic organisms floating in the water. Many of these organisms stick to the mucus layer and are transported along with the mucus into the stomach. The water passes through the gill slits, these openings here, into the atrium. The atrium is a chamber that surrounds the pharynx on three sides. You can observe a portion of it here beneath the pharynx. Here is a view of the rear portion of the digestive system. On the left, you can see the pharynx with its gill slits. Water passing from the pharynx moves posteriorly through the atrium, ultimately exiting through the atriopore. Food trapped in the mucus within the pharynx is carried into the stomach. Here the process of digestion and absorption begins, aided by enzymes that are secreted by the hepatic cecum, which I will show you shortly. Digestion and absorption continue as the food moves through the intestine, and any undigestible material passes out through the anus. Here is a view of the middle of the lancelet, in which a portion of the pharynx has been removed. This allows us to see the hepatic cecum, which is attached to the stomach. This blind-ended pouch secretes enzymes and takes part in the absorption of nutrients. Evidence indicates that this structure is homologous with the liver in vertebrates. The blue structures visible behind the hepatic cecum are gonads. They may be either testes or ovaries. Now we will look at a cross-section of a lancelet. This slide is available in the laboratory. This circular structure is the notochord. Just dorsal to the notochord is the nerve cord. And these large stringy areas of tissue are myomeres. That is, they are the muscle. This circular structure is the pharynx. The middle is the lumen of the pharynx. These dark structures are the gill bars, and the lighter regions between the gill bars are the gill slits. The white space surrounding the pharynx is the atrium. To make sense of the other structures in the cross-section, let's look briefly again at the model. This dotted line indicates the approximate location of the cross-section of lancelet that you are seeing. Note that a portion of the pharynx is removed, revealing the hepatic cecum and the gonads in the model. Looking back at the cross-section, this structure to the left of the pharynx is the hepatic cecum. The large dark staining structures here in the lower left and in the lower right are the gonads, in this case ovaries. Now, Let's take a brief look at the other subphylum included in the Protochordates, the tunicates, which are in Urochordata. Once again, your textbook has useful information that will help you in your laboratory studies. Looking at the diagram of an adult tunicate, it may be unclear why these organisms are included in the phylum Chordata, since they appear to lack 3 of the 4 derived characters of the phylum. However, the diagram of the larval form of the organism reveals all 4 of the expected characters. The adults, being sessile, which means permanently attached to a substrate, lose unneeded structures, including these 3 derived characters. Here is a whole mount slide of a tunicate that is available in the laboratory. It has been treated chemically to make internal structures visible. Adult tunicates are permanently attached to a substrate and gather food by filter feeding. Water is drawn into the organism through the incurrent siphon by the beating of cilia. Water flows into the pharynx, which, as you can see, occupies most of the body of the tunicate. There are no obvious gill slits. Rather, the pharynx resembles a densely woven net. As water passes through this net, planktonic creatures adhere to the mucus coating. The water enters the atrium, which surrounds the pharynx on all sides, but is most easily visible here on the right. Water exits through the excurrent siphon. The food-laden mucus, which is within the pharynx, is carried down to the digestive system for processing. As always, any undigestible material is expelled through the anus. This, as you can see, is located near the excurrent siphon, for obvious reasons. That concludes our look at the specimens and models of lanceolets and tunicates that are available in the laboratory. I hope this helps you to prepare for the upcoming laboratory practical.